Good afternoon. Welcome everybody to the last seminar of this Young Investigator edition. First of all, I would like to thank Dr. Jordi Gracia Sancho and Dr. Rafael Bañares for giving me the opportunity to be the host today. This afternoon, the study is Dr. Jeremy Gauthieron, and after the presentation, we will have the pleasure to incorporate Professor Ratziu and Professor Romero Gomez into the discussion and the debate. Now, let me introduce you to Dr. Jeremy Gauthieron. He is a full researcher at the Sorbonne University and in CERN at Saint Antoine Hospital in Paris, France. His group aims to characterize the initiation, regulation, and molecular function of regulated cell death in obesity and related disorders such as non-alcoholic steatohepatitis and type 2 diabetes. Recently, they have published a relevant study showing that the use of RIPA56, a highly specific inhibitor of RIPK1, represents a promising target for the treatment of NASH. Now they are working on bringing the RIPK1 inhibitor into the clinic. Dr. Gauthieron graduated in biological science at the University of Pierre and Marie Curie in 2008 and received his PhD in cellular and molecular biology from the University of Paris. This was followed by a postdoctoral work at the University of Aachen in Germany, and during that time he worked intensively on the TNF signaling pathways, studying the role of NF-κB and programmed cell death pathways in liver disease. The presentation is entitled Necroptosis in NASH, a new therapeutic target, and I am sure that it's going to be very provocative. Jeremy, thank you very much for being here today and accepting the invitation. And please, whenever you want, it's your turn.
So thank you very much for the kind introduction. So I'm very glad to be with you. And of course, I'd like to thank the organizer and Javier for the invitation. So as he told you, so I'm going to talk about necroptosis. It's a novel form of programmed cell death in non-alcoholic steatohepatitis. And I try to highlight the therapeutic potential of necroptosis in NASH. So what is NASH? I'm sure that everyone knows, but it is the advanced form of non-alcoholic non-alcoholic fatty liver disease. I will try to take this one. And within the spectrum of the disease, this disease, you can find different entities. So the first one, let's say it's non-alcoholic fatty liver. It is mainly characterized by a steatosis in the liver, and it has to be like 5% of total hepatocytes that are full of fat. And of course, inside the stage of this disease, you can find different degree of steatosis, and it can go to 90% of steatosis, of course. And the second stage, I would say that it's non-alcoholic steatohepatitis. So it is still characterized by steatosis, but it causes inflammation and hepatocellular death. And both events can lead to scarring of the liver, and you can develop fibrosis. And I would say this stage is NASH plus fibrosis. And when you are in this stage of the disease, you can go further to cirrhosis and cancer. So NAFLD is closely related to obesity and metabolic syndrome. And for this reason, it appears like a few, let's say 1 or 2 years ago, the researchers started to think about another name, and now it tends to be named MAFLD for metabolic associated fatty liver disease. So you can find also this term to define NAFLD. And NASH is predicted to continuously rise in the future and to become the most common indication for liver transplantation. And my group tried to understand the mechanisms, the molecular mechanisms responsible from the transition to NAFL to steatohepatitis. And as you can guess, one of our focus is hepatocellular death. So to introduce cell death, I'm going to talk about external death activation pathway, and it is driven by death ligand like TNF, so tumor necrosis factor, and it basically is the same with other death ligand like FAS or TRE. And the binding of TNF on its receptor will trigger a signaling complex, this one. And as you can see, there is many proteins inside, so you can find adapters like TRAD, FAD with a death domain for the association with the receptor. You can find E3 ligases like TRAF2, SIAP1 and 2, also kinases like RIPK1, deubiquitinases like LUBAC. And as you can see, it's highly regulated by post-translational modification, mainly ubiquitins. And I won't go through details, but every component of this signaling complex are modified by ubiquitins. So you can find everything what you want to look for. And basically, you have to retain that it activates the IKK complex with several subunits. One is a regulatory subunit called NEMO, and two others are kinases, IKK1 and 2, or IKK alpha and beta. And this IKK complex phosphorylates the inhibitor of NF-kappa B, and this inhibitor goes to proteasomal degradation. And NF-kappa B is free to enter inside the nucleus and activate its target genes. And within the target genes, you can find genes regulating cell survival, proliferation, cytokine production, and therefore inflammation. But NF-kappa B is not the ending story of this death ligand pathway, because most of the time you can have another switch towards different complexes, which lead to the activation of caspases, and so to the activation of apoptosis. And so I just put some video just to show you. So you will see, of course, that the main feature of apoptosis, it is this shrinkage of the cells. So there is the condensation of the cytoplasm. And what you have to get in mind, because I'm sure you know that it's a non-inflammatory cell death pathway. I mean, the plasmic membrane are preserved, so you don't have release from everything what's inside the cells. It does not go outside, and one of the features is also the apoptotic bodies. And you can think about a macrophage going on in these dead cells and just swallow the dead cells, and nothing happens more than that. And of course, everything is solved afterwards. I think recently, since it was like that, there was like such a balance between death and life. And I mean, if the NF-kappa B stimulation was sufficient, I mean, it could counteract apoptosis via, for example, anti-apoptotic genes, and therefore the cells could survive. And if it was the opposite, the stimulation, apoptotic stimulation was, I mean, increased stronger than the NF-kappa B stimulation, then the cell will die. But once again, it's not the ending of this story, because it was like in 2009, so it's now more than 10 years, researchers started to manipulate this apoptotic pathway. And they found out that by removing caspase-8 chemically with the use of an inhibitor, or by removing it from cells, you've got a switch from apoptosis to a new form of cell death, new form of programmed cell death, named necroptosis. So necroptosis is driven by two kinases, RIPK1 and RIPK3, and a pseudokinase named MLKL. And they form what we call the necrosome complex. And within, RIPK1 phosphorylates RIPK3, RIPK3 is active, then it phosphorylates MLKL, and MLKL can execute necroptosis. So how does it look? So I put this video for you, so it's quite fast. So you can focus on this cell here. So you can look at this one, and now this one. So I think you have seen that it's completely different than apoptosis. It looks like necrosis. So you have the swelling of the cells. So you have like an explosion. And what you have to take in mind, it is necrosis, but it is programmed. You need a death receptor. You need somehow an inhibition of caspase-8 chemically by removing it from cells, or also physiologically. I cannot say this word, but it can happen naturally. And at the end, MLKL goes to the plasmic membrane. It makes some pores. And so you have this osmotic shock. The water goes inside the cells, and you have this explosion. So everything goes outside the cells. So it's highly an inflammatory process of death. And of course, it has been linked to many diseases now. And you can look at this disease, for example, on the left. So it's, let's say, a gastrointestinal disease. You have also now neurological disease, skin inflammation, the cardiovascular axis with stroke, ischemia, reperfusion injury, also atherosclerosis. Of course, necroptosis is involved in infection, bacteria, viruses. And in the lab, we are really focused on liver, of course, with NAFLD, but also on the adipose tissue. And just because NAFLD is linked to obesity and type 2 diabetes, so this axis between two tissue organs are really important for me. But I will mainly show some result on liver. Okay, so what is known about NASH and apoptosis? So it was in 2003, so the group of Feldstein started to investigate what was going on. And they could find out that there was some positive cells, tunnel positive cells in a NASH patient. And they also tried to make some staining for CLIF caspase 3 and 7, so effectors caspases in human. And okay, it looks a bit unspecific, but as you can see, there is like a brownish stain. So probably there was apoptosis going on in this patient. And in fact, in 2011, there was a clinical trial with the pan-caspase inhibitor. It was named GS9450. And Vlad and colleagues could demonstrate that there was a reduction of transaminases in this NASH patient when you use the pan-caspase inhibitor. But it was like a small study. It was not too long, the treatment. And but at least there was something going on. So probably in NASH, apoptosis was activated. And using a pan-caspase inhibitor, you could reduce the rate of liver injury. So what about necroptosis? So we started to investigate necroptosis and NASH in 2012, let's say, after the study of Vlad, the clinical trial. And for that, we use the methionine and choline deficient diet. So it's a feeding model for mice. And when you do that, when you fed the mice with this MCD diet, I mean, the mice, maybe after 2 weeks, they start to develop steatohepatitis. It's really fast model, so you can basically find out if it's involved or not really rapidly. So we took some mice, so wild-type mice, and we look at the expression of RIPK3. And you can see here the basal expression of RIPK3. And when we fed the mice with MCD, it was quite increased, as you can see here. We take out caspase-8 from the liver. specifically from the liver. So remember that it's one of the break of necroptosis. If it's present, it can cleave RIPK1 and RIPK3 to prevent necroptosis activation. And when we take out from the livers, as you can see, the basal level of RIPK3 is already high. And when we fed the mice with MCD, so we just developed steatohepatitis in mice, the level of RIPK3 is even more increased. So you can, of course, you don't see RIPK3 in liver when you take out RIPK3 from mice. And when we look at caspase-8 levels, it was not regulated. Okay, does it induce liver injury? Yes. So this is transaminases reflecting liver injury. So this is wild-type mice under feeding. So you see that a mild expression of RIPK3 induced liver injury. It was even more when we block caspase-8. And you can revert the phenotype by removing RIPK3. So double knockout mice were somehow protected, and the single knockout mice were protected. Look at the difference with wild type. It's quite obvious. So it was not reduced at the minimum level. There was a bit of damage, but I mean, it was quite reduced. So we could look at immunohistochemistry for RIPK3. So this is control mice, wild type mice under normal show diet. This is MCD diet mice. So when you take wild-type mice, you see that hepatocytes were positively stained for RIPK3, as you can see here. Maybe there was also other cells that were positively stained. When you remove the brake, so necroptosis is just free to go, and there is no caspase-8 anymore. Look at what we observed. It was really full of RIPK3. It was not only hepatocyte, but it was really everywhere. Everything was positively stained. You could revert the phenotype of caspase-8 LPC knockout by removing RIPK3, and single knockout, of course, you don't see any staining. So we could correlate this with inflammation. So wild-type mice, a bit of RIPK3, we could see like macrophages. They were organized into foci. It was looking a bit like cone-like structure that you can see in adipocytes. And in this field, for example, you could see 1, 2, 3, 4 foci of macrophages, maybe reacting to cell death, to necroptosis. And when you remove the brake of necroptosis in these mice, you can clearly see that the foci number were increased. And I mean, in this field, it was like 10 to 12. And for the double knockout mice or the single knockout mice, you could see macrophages, but they really looked like normal mice. They were not organized within this foci. And you could correlate with fibrosis, RIPK3 a bit of inflammation, mild fibrosis, lots of RIPK3, lots of inflammation, lots of fibrosis, and no fibrosis at all for the double knockout mice or the single knockout mice. So at that time, we could look at NASH patients, and it was a small cohort of patients. It was biopsy from NASH patients, and as you can see here, the result was quite clear. In this biopsy, we could see an overexpression of RIPK3. And when we look over apoptosis activation, CLIF-Caspase-3, it was not so clear. And in this blood, it was even a small amount of CLIF-Caspase-3 compared to controls. We could look at in immunohistochemistry. It was not so beautiful at that time, but I mean, for one patient, we could have like biliary cells that were positively stained. So it was like something was going on in biliary cells. We could have this funny granule-like staining pattern. So I don't know if it was really specific, but what I can tell you now, the necrosomes, so RIPK1 and RIPK3 can associate and can form amyloid-like structure. So maybe it reflects this kind of structure really condensed, really huge amount of protein, and maybe you can visualize them, but I'm not sure, of course. But what was really funny, you could find in patients like zone of hepatocytes positively stained for RIPK3, so maybe if you count, it was like 10 hepatocytes. And surrounding, you could find all these immune cells. So it was macrophages, T cells, B cells. You could make some double staining and you could find everything. It was like these hepatocytes were dying and immune cells were reacting to the inflammation and were close to this zone to do something, to react on this death. And it was 1 year later, the group of Professor Rodriguez just confirmed these results. So, and add also other results, and they could demonstrate that necroptosis was a key pathogenic event in human and experimental murine model of NASH. So it was nice that other group could also show some stuff like this. And I think we should be able to publish and to confirm some results. And it's a bit a shame that it is so difficult to publish and to confirm results from others. And you always need to add lots of stats, lots of results, findings. And I feel a bit sorry because, I mean, it's important that the scientific community can confirm results, and like this we can move forward and we can just make better science. And the story is not the end here, because there was a novel clinical trial with pan-caspase inhibitor. It was last year in Journal of Hepatology. And it was this time with Emricasan. So it's a pan-caspase inhibitor. And it was in a patient with NASH plus or minus fibrosis. And the conclusion was the following. Pan-caspase inhibition with emricasan did not improve liver histology in NASH and may have worsened fibrosis and ballooning. So it was even more, it was a bit like what I have showed you with mice. I mean, you remove caspase, so here it's genetically, but you could also use a pan-caspase inhibitor and you switch to necroptosis. And this was one conclusion of the paper. While speculative, pan-caspase inhibition may have switched cells from apoptotic to more inflammatory form of cell death. So of course, they did not name it necroptosis, but I could bet that at least there was necroptosis going on, or maybe also some kind of others. It could be pyroptosis or maybe ferroptosis or something else. But you inhibit caspase, you have this switch towards inflammatory death, and it's not good for patients. And for the story, because it was before, and with this pan-caspase inhibitor, so the first one that was used in this preliminary study with clinical trial in NASH patient, so it was GS9450, and it has been tested in patients with hepatitis C before we get the treatment, of course. And the clinical trial was stopped really fastly for adverse effects. And of course, this kind of clinical trial, when it's liver, the readout is always transaminases. And I could bet that the transaminase went high with this pan-caspase inhibitor just because it's speculation. We don't know, and probably we will know not the clue of it. But I mean, viruses activate necroptosis, and if you block caspases, I mean, you will switch toward necroptosis, no break at all, and it can go full, and of course it will make transaminase liver injury in hepatitis C patients. So definitely, pan-caspase inhibition, I think, is not the good option. So apoptosis inhibition won't work in NASH patients. So far we are here. So hepatocytes, fat accumulation drives injury. You have some dead hepatocytes probably, so apoptosis a bit, probably necroptosis or other kind of cell death, unidentified or not so much involved, but inflammatory cell death. It induces a primary inflammation via DOMS, cytokines, maybe extracellular vesicles. It also has been shown maybe they play a role, and it activates Kupffer cells. And this Kupffer cell will recruit a monocyte from the blood via MCP1, and the monocytes will become inflammatory macrophages. It will lead to a secondary inflammation, leading to more cell death. And here we are in a vicious circle. So death, not only hepatocyte, but also probably inflammatory macrophages, because I mean, it's full of, they probably will react also and die. And of course, more death, more inflammation. And the idea really is maybe, of course, to block death, but to block this primary inflammation. And at that time, I was wondering if necroptosis inhibition may work, and maybe because it acts on cell death, but also it will remove this primary inflammation, maybe it will be beneficial for NASH patients. So we decided to test this in a murine model of NAFLD. And how can we inhibit necroptosis? So this is an example of in vitro with cells. So these cells are a bit special because you put TNF and they can die. So you can look at here, they die just with TNF. You don't need to inhibit caspase-8, but they die by necroptosis. And if you block RIPK1, a kinase activity of RIPK1 with RIPA56 or necrostatin, it's the first drug that was discovered for inhibiting RIPK1 kinase activity. But as you can see, you completely rescue the death by the TNF. And when you look up the phosphorylation of MLKL, it's also abolished. I mean, you don't have any more phosphorylation. So inside the necrosome complex, by inhibiting the kinase activity of RIPK1, you block the phosphorylation of RIPK3. So RIPK3 is not active anymore, so it cannot phosphorylate MLKL, and so MLKL cannot activate necroptosis. So that's it. So you can give the drug to the mice and see what's going on. So this is the work of my PhD student, Amin Majdi. So we took a model of mice that is closer to human. So it's high-fat diet. So it's lots of fat inside the diet, but you have also high sucrose inside. And you can feed the mice for 4 months, and it will develop NAFLD. So it's less stronger than MCD diet, but at least if you go for 4 months, you will see a bit of inflammation and also a bit of fibrosis. And another group was with RIPA56 in the food, and it was the prophylactic settings. So it means that we give RIPA56 in the same time when we started the feeding. And another group, we just leave the mice 3 months under this feeding, high-fat diet feeding. So they become obese, they develop NAFLD, and then we decided to treat them for 1 month. And we just keep going high-fat diet, and we treat them with high-fat diet. So when we look at the weight of the mice, so we were a bit surprised because when we give the inhibitor, the kinase inhibitor for RIPK1 in the same time that the high-fat diet feeding at the beginning, so as you can see here, there was like a reduction of obesity. So it was not so strong. It was like 20 to 25%, or maybe 20% more than 25, but it was significant. So there was something going on, but the mice were still obese. Like maybe look at it was like around 40 grams, when a normal show diet mice is around like maybe 25 grams after 4 months. And when we look at the group of the mice with the therapeutic strategy, so we gave the inhibitor just 1 month here. you see that there was not so huge impact of obesity, and the mice were quite similar in terms of weight than the high-fat diet group. And it was around like 45 grams to 50 grams, it depends on the mice. So we just keep going with that, and we look at if the inhibition of RIPK1 may reduce liver injury. So you can look at first at this blot. We look at the expression level in liver of the necrosome component. And by inhibiting RIPK1 kinase activity, I mean, we do see some variation, but RIPK1 was still present in the liver. So maybe it was a bit strong, overexpressed a little bit when you compare to high-fat diet. So probably it may stabilize RIPK1, and the half-life was increased. And when we look at MLKL or RIPK3, as you can see, it was clearer, and we could see a strong reduction of the expression of MLKL and RIPK3. And when you look at the readout of liver injury, it led to a reduction of transaminase, as you can see here. So this is high-fat diet mice. So you have mild injury, like it's around 200, and the injury is completely gone when you use in the prophylactic settings the inhibitor, and it's also quite rescued for 1 month of inhibition. When we look at inflammation in high-fat diet, you could find some area where there was macrophages around hepatocyte. Again, this kind of foci activating macrophages. And when we look at the mice with inhibitors, it was not so obvious. There was sometimes some foci, but it was not as strong than high-fat diet mice. Afterwards, we look at fibrosis, of course, and as I told you, it's not the best model for fibrosis, but I mean, when we quantify it, the pericellular fibrosis, we could find something like around 2% of the liver. And when we compare to both settings with the inhibitors, it was not anymore there. But we look up collagen expression. And as you can see in high-fat diet group, so it's highly increased, and this increase is completely gone with the prophylactic settings. I mean, it's like a normal show diet, and it already reduced by 25% when you use 1 month of treatment. So this was, I mean, kind of expected results. Inhibition of necroptosis reduces liver injury, so transaminase readout, inflammation, macrophages, for example, and fibrosis with collagen. And this was, I mean, what we expect. And it was not a surprise at all. And other people tend to show some result like that using necrostatin instead of RIPA56. But what was really interesting, this is these results. I mean, look at high-fat diet group. You can see like it is full of steatosis. I mean, it was like 100% of hepatocytes. So you could see like macro steatosis, but also micro steatosis. It's the same word for me, for a French guy, but I mean, you understand what I mean. So big vacuoles, and like these big vacuoles, they cannot be sustained by the hepatocytes, so they are cleaved in several vacuoles, and they make this ballooning of hepatocytes. And look at what we have seen with the prophylactic settings. So at the beginning, no, I mean, it is clear, no steatosis, or almost not that much, and it's really surprising. And I remember that the mice are still obese, 40 grams. So it's reduced, but they are still obese. And look at 1 month of treatment. This was like amazing. I mean, the differential is insane. And you could quantify the score, the steatosis score. So it is there, so it's highly reduced. a bit less for the curative settings. And you could also quantify triglyceride inside the liver. And I mean, high-fat diet, it's 100%, and you have a reduction in both settings around like 50%. So this was the conclusion. So beside activation of cell death, the mediators of necroptosis may play a role in metabolic pathway. So is it accumulation of triglycerides? Is it the use of triglyceride? That was the next question we try to figure out. But just before that, we wanted to test these drugs in a physiological context. And we had the chance to have the liver graft from NASH patients. So those patients went for liver transplantation, so we could get access to the liver. We put the hepatocytes in cell culture. And as they are already from NASH patients, you don't need to do anything else. They are already full of fat. They are loaded with fat. And so you can basically just treat them with DMSO, so the vehicles of the drug, or RIPA56. And I think it's quite clear. The picture is quite clear. When you do that, you have a reduction of the staining, but also the triglyceride that you can quantify inside the hepatocytes. And it was like 25 to 30%. And these experiments were done overnight, 16 hours. So we put the inhibitor 16 hours and you have 25% of the triglycerides that are vanished. So we could repeat the experiment using another inhibitor because we wanted to be sure that it is related to RIPK1, but maybe it would have been related to MLKL, the downstream targets. So we took primary hepatocytes. This time they were not from NASH patients, so we needed to load them with free fatty acids. So we did that. So you can see the ratio without free fatty acid and DMSO. So it's much more. And we treat them with NSA. So it's a specific inhibitor of MLKL, human form. So we cannot use it in mice, unfortunately. And when we did that, I mean, it's quite spectacular too. So we had a reduction of 25% of triglyceride within like 16 hours. So what does it mean? It means that it's related to the necrosome complex, and the main molecule that makes this defatting process or this metabolism stuff or react, it is MLKL. It is the downstream targets of RIPK1, the main effector of necroptosis. So we went a bit further and we took away MLKL from this hepatocyte, murine hepatocyte, by using CRISPR-Cas techniques. So you see that the knockout is highly efficient. So you can put them in culture, so you don't see any differences between basal cells. And when you load them with free fatty acids, I mean, there was a differential around 50% of accumulation of triglycerides. So we could quantify it and we could also look at triglycerides. So MLKL controls somehow triglycerides in hepatocytes. So accumulation, defatting, this we didn't know yet. And by removing it, it's better for the cells. It's better for hepatocytes. And you can just remove or also use a pharmacological inhibitor and you have the same effect. So we put these cells in a seahorse analysis because we wanted to see the respiration, mitochondrial respiration. And so I won't go through details, but you can see already the differential between the knockout mice, the knockout cells, in blue and the wild type cells. You see that the basal respiration is already increased over like maybe 50%. The ATP-linked respiration was also increased, and the maximal respiration, the maximal capacity were also highly increased. So I won't go further in details, but this is the model we initiated. So RIPK1 activate RIPK3, RIPK3 activate MLKL. MLKL has two phases. One is towards cell death, so it promotes hepatic cell death. So hepatocyte will die by necroptosis, it will promote inflammation, fibrosis, and it will make steatohepatitis plus fibrosis, of course. But on the other side, it's also prevent the mitochondria to make, for example, beta oxidation. They are less good. They are less good to use triglycerides for making energies. And instead of, as they are somehow inhibited, they will make, the cells will just store triglycerides, storage of triglycerides. And of course, it will increase steatosis, and steatosis will contribute to make NASH because it will also induce death in hepatocytes. And when we use an inhibitor of RIPK1, kinase inhibitor, so you block RIPK1, so there is no phosphorylation anymore to MLKL. So MLKL is not active, so it cannot make death. And the mitochondria are free to work, and they work better, and they can use triglycerides, and they can make energy, and instead of the storage. And it was the same time than us, like I think the same issue, or the group of Professor Nagy in United States. They make also nice work out of it, and they could use the knockout mice with MLKL, and they use this high fructose, high cholesterol feeding. And as you see, the wild type mice, the negative wild type mice. Oh no, this was full knockout. So the wild type mice were full of fat once again. And look at what they observed. I mean, the knockout mice fed with FCC diet. I mean, there is no more fat anymore. There is nothing. And they could link MLKL this time with autophagy. So maybe there is something going on with mitophagy or lipophagy and the use of these triglycerides. So, I mean, when you look at therapeutic targets in NASH, so I put few of them here. So most of them are, I think, failed in clinical trial, like the pan-caspase inhibitor, ASK, targeting this axis of the disease, they failed. CCR2, CCR5, I'm not sure, so maybe we can ask later, Vlad, if it's also cancelled. PPAR agonist did not work, but they target only the metabolism axis of the disease. I think FXA agonist is ongoing. But I was thinking, when I look at this different axes of the disease, that maybe necrosome inhibition might be beneficial in patients because it targets two main axes of the disease. So stress oxidative with cell death, but also steatosis and metabolism. So it is like, it has pleiotropic effect. We could show it in mice. It reduces liver injury inflammation, but also steatosis, also in human cells. So RIPK1 inhibitors show beneficial effect not only in prophylactic settings in mice, but also in curative settings. And I think it's really important because most of the drugs before were tested in the context of prophylactic settings and not in the context of curative settings. And I think it's important to do that in future for new targets. And of course, the inhibitors, RIPK1 and MLKL, works on primary human steatotic hepatocyte directly derived from human graft of NASH patients. So I think I was not too long, and so I'd like to, of course, make some acknowledgement, and I'd like to thank Professor Chantal Housset. So I'm in her department, and of course, she gave me the opportunity to develop my own group and give me lots of support. So this is my group, so the past student with Amin and Tayeb, and my group with my new PhD student, Julie, technician Celia, and some MD and master student, Pierre-Antoine and Sarah. I'd like also to acknowledge other members. from the lab or not from the, or also outside. So I put them here, and they give me lots of support in my research. And of course, I have a special word to my collaborators. So first of all, Professor Vlad Radziu. I mean, I came back France, and the first things I've done, it was to contact him, because I absolutely wanted to work with him. And he gave me lots of support for everything. So I'm really grateful. I'd like also to acknowledge Professor Bruno Febvre and Isabelle Giroux, because as I told you, I will also work on adipose tissue. And in this field, we are also proactive. We have several papers on revision, and without them, it won't be possible. I'd like also to thank the hepatobiliary surgery department. I mean, Professor Olivier Scaton, Claire Goumard, Philomena and Linda, because they gave me also lots of supports. We work on this transplantation aspect. They give me like human samples from NASH patients, from graft. It's amazing what they have done for me. Also, the Portugal with Cecilia and Marta. I mean, I also contacted them when I started my group, because I mean, they are fantastic when we do lots of work together on metabolism with K3, MLKL, all this stuff, and nice stories are ongoing. And to finish, I'd like to also thank Australia, Dr. Kim Bridle, and a fantastic PhD student, Rajiba India. And we work together on ischemia reperfusion injury in steatotic livers. And we have also some good story ongoing. And of course, I need to thank the funding agency, and because I need them for the support, for the financial support, and without them, it won't be possible. So I'd like to thank the Centre de Recherche Saint-Antoine, Fondation pour la Recherche Médicale, Ville de Paris, Société francophone du diabète, and ICANN. It's Institute of Cardiometabolism and Nutrition, and thanks for their support. Thank you very much for your attention, and I'm pleased to take any question. And I hope you have enjoyed this talk.
Fantastic presentation, Jeremy. I think you have shown a cutting-edge frontier in NASH therapy. Before moving to the questions, I would like to greet Professor Radziu and Professor Romero Gomez and invite you to participate. Briefly, Dr. Vlad Radziu is Professor of Hepatology at the Sorbonne University and the Pitié-Salpêtrière Hospital in Paris. He completed his medical training at the University of Paris, a postdoctoral fellowship at the Liver Center at the University of California in San Francisco. And finally, he earned a doctoral degree for his work on the pathophysiology of viral and metabolic liver fibrosis. His main research interest is NAFLD, including pathophysiology, risk factors, and progression of liver fibrosis, as well as the treatment of NASH. He coordinated the FLIP consortium, and also he's part of the steering committee of the Horizon 2020 NASH EPOS and the Litmus consortia. And currently, he is a co-editor for the Journal of Hepatology and a member of the International Advisory Board of the Lancet Gastroenterology and Hepatology. On the other hand, Dr. Manuel Romero Gomez is full professor of medicine at the University of Seville, head of the Digestive Disease Department at Virgen del Rocío University Hospital, and head of the Group for Translational Research in Liver Disease at the Institute of Biomedicine of Seville. He coordinated in the past the Spanish Registry of NAFLD-NASH, EPAMED, and is the co-coordinator of the EASL European Registry of NAFLD. Also, he's participating actively in NASH-related European projects such as FLIP, EPOS, and Litmus. And both Vlad and Manuel have published more than 200 papers in top 10 journals. So again, welcome. And now we can start with the questions.
Yeah, that's the point. I think it may have a potential value by reducing adipose tissue size or stuff like this. It was what we observed in mice. I mean, the adipose tissue was reduced, so that's why the weight was lesser than wild-type mice. So of course it's a good point, and this is what we try to investigate. We are looking up if the necrosome play a role. I mean, reducing inflammation, maybe adipocyte differentiation, all this stuff. We are working on it just to look at what is going on. It's a good point. And of course, it is beneficial. Sugar, you mean? Yeah, we have a bit, and up to now, I don't know the switch, and I don't know if the differences, but of course, I can't tell you more because we did not so much investigate it. But for sure, what we used for the mice, it's called high-fat diet, but when you look at the component of this diet, it's also full of sugar. It's not pure high-fat diet. You have also plenty of sugar. So probably the pathway, the lipogenesis de novo is maybe inhibited or some stuff like this. And we are working on this field also with Cecilia to look up what is going on. So for RIPA56 treatment, no, we did not see. But for sure, when you target RIPK3, so it's quite different. And most of the time when targeting RIPK3 will trigger apoptosis. But it's less true when you target RIPK1 or MLKL. See, it's more related to RIPK3. Somehow, by inhibiting RIPK3, you switch to apoptosis. That's why you cannot use this inhibitor for human, for sure, because it's also disbalance death towards apoptosis. But RIPK1 and MLKL is somehow different, and it does not make sense. And I mean, there was this phase 1A or 1B using a necroptosis inhibitor in human just to look at toxicity. And I think with the normal dosage, they did not see any elevation of transaminase. Of course, when they give much more, maybe they see a bit, but like every drugs, I think when you give an overdose or something, it's always toxic for liver. But I mean, it was well tolerated for human. So we did not see any switch. Definitely, for me, having seen 2 clinical trials with adverse effect with pan-caspase inhibition, I mean, for me, definitely, we should forget about inhibition of apoptosis in NASH. In context of fat, at least, I think it won't work. So you switch towards something, and I mean. There is still this game. We don't know the exact trigger of necroptosis, and you can imagine like a triglyceride or a saturated fatty acids, or can directly activate necrosome. And I mean, in this context, this specific context, I think inhibition of caspase, because it's a pan-caspase inhibitor, and I think we don't have specific inhibitor, it will be a bad solution, and you will immediately switch. Because I mean, probably necrosome is primed inside the hepatocytes, and I mean, they are ready to die, and they are blocked by caspase or other stuff. And I mean, you dysregulate the activity of caspase, and boom, explosion, and it can make the damage. So I won't use it personally, but Vlad maybe can say some word about it.
Yeah, no, first, thank you, Jeremy, a beautiful presentation. For the first time, I start to understand the whole process, because you explained it really well. I don't know specifically, we have to look at the data, and the data has shown 2 negative studies. So there's probably not the best way to tackle NASH. I would have 2 simple questions, and maybe you already explained that in the beginning, but just maybe for people to understand, particularly those that are not really specialists in that. So when you look at apoptosis, there are markers of apoptosis that you can use in histology to demonstrate that some cells undergo apoptosis. You can do tunnel, and you can identify apoptosis going on, like the paper by Ariel Feldstein that you have shown in the beginning on your slide. Is there anything similar for necroptosis? Can you look at a slide, use some immunohistochemistry marker, and demonstrate the presence of necroptosis? And I have another question after that.
Yes, this is a really great question. I mean, you can definitely you can look at phosphorylation. So phosphorylation of MLKL, and basically when it's phosphorylated, normally it activates necroptosis. But there is also some papers showing that you can have phosphorylation without execution of necroptosis. So it's quite difficult up to now to really define and to really look at necroptosis in histology. And also the tools are a bit not good enough, like a phosphoantibody that you can find commercially are not good. And then these phosphoantibody are trusted by big companies. And now they tend to sell it for researcher. But I mean, the last 5 years, it was quite difficult. But now something is going on, some specific antibodies for phosphorylated form, like special residue, like serine or whatever the position, they are coming, and obviously some of them are specific to necroptosis activation. So soon we can define it. And tunnel positive cells, so it has been used for apoptosis, but I have to say that also necroptotic cells or necrosis cells are also positive for tunnel. So at some point, so I mean it's clearly not specific enough for apoptosis.
Okay, so it's hard to tell. So then I have two things that are related. So the first one is to try to understand if we can somehow quantify the amount of cells that undergo necroptosis. Is it something really a minor event happening in the liver, or does it explain the bulk of hepatocyte cell death in a disease such as steatohepatitis? Strangely enough, when you look at the apoptotic cells using these immunohistochemistry methods, might not be perfect, you see here and there some apoptotic cells, but it's not like massive apoptosis like you could see in acute injury. So then you always ask yourself, is that really what matters for inducing inflammation? So the first question is, is there a way to sort of have an idea of quantitatively if this phenomenon is predominant or if it's just anecdotal? The second thing is, okay, it's a way for cell death, but does it induce inflammation? Does it induce particularly, for instance, ballooning after that? Maybe one thing to do in the future, I know you've shown something about inflammation, but maybe you can develop a little bit and say how a necroptotic cells signals for inflammation. And second, since ballooning is such an important issue, there are at least one marker that engages the cell towards a balloon phenotype, which is the sonic hedgehog signaling. So maybe one thing to look at in this necroptotic process is whether that triggers sonic hedgehog, for instance, so that you can link this to features of steatohepatitis more directly.
Yeah, this is a good point. So we have to do it with sonic hedgehog for sure. I mean, I cannot answer this question. It's a good question. So I mean, this is what everyone wants to do. I don't know if it's anecdotic. For sure, apoptosis maybe. The way that you can find small dots in the liver of NASH patient, maybe it's reflecting the fact that the apoptosis it's better than necroptosis. It just reflects this disbalance. It's maybe better to die by apoptosis than necroptosis. But of course, this is what we said just before. We need better tools to evaluate, to quantify necroptosis in NASH patient and to see what is going on. And of course, the study of Arun and in JEP, it would be nice to have like a serum or all this stuff, serum, for example, and to look for necroptosis inside markers, because if it's dying, if they are releasing, if it's necroptosis death, everything goes outside, and you can find like markers of necroptosis inside the serum of patient. You will find like RIPK1, a phosphorylated form of RIPK1, RIPK3 MLKL. It will be released, and it will indicate if the amount of death in the liver. And this is the readout we have already. So you make some ELISA in NASH patient and you see that it increases in the serum. So it indicates that the amount is quite important. But as I remember, it was the same with apoptosis. So it was also in the serum of NASH patients. So I don't have the clue. I don't have the answer yet. But I mean, it is what we have to do. We have to put some effort to evaluate death. And some people started to overview the death as a pan death. So I mean, they said like in NASH patient, probably you have apoptosis, you have necroptosis, you have ferroptosis, iron-dependent death, pyroptosis. You have a combination of all the deaths, and this trigger the damage. So I would say that maybe you have a bit of apoptosis, you have a bit of necroptosis, you have a bit of pyroptosis, and you have a bit of ferroptosis, and all of these events combined make lots of cell death and will promote NASH. This is the way I see it. And of course, necroptosis inhibition, you could say like, look, it won't target iron-dependent cell death or pyroptosis, but it also has some link with them. And for example, it has been shown that MLKL regulates inflammasome. So by taking out MLKL or by inhibiting MLKL, you may have a positive link with pyroptosis and inflammasome activation. So I mean, when I see it, I see that it has only beneficial effect. Maybe not on iron, on ferroptosis death, but I mean, it will act also in pyroptosis, inflammasome, so it might be beneficial. But we have to overlook everything. We have to quantify all the deaths and see the quantity of it, and maybe it will be huge, and it will explain why steatosis is going on. I mean, ballooning, it's believed, the hepatocytes are believed to be like somehow not working anymore, like inert. They don't do anything. But I was wondering, for example, if microvesicles can still be released. You could imagine like you have vesicles going outside full of MLKL or full of stuff, because remember that MLKL goes to the membrane plasmic. They form some pores. And it has been shown that sometimes it can signal without making death. So I was wondering if like a senescent cell or ballooning cells can still release these microvesicular vesicles full of MLKL, full of everything, and it will signal outside surrounding hepatocyte that may react and die or make inflammation or stuff. So I think it's a good question. So it's like you, I don't know. I think it's a consensus. No one knows exactly, but there is these parts, tricky parts with vesicles that might be released by ballooning cells, and it might play a role in the pathology. Yes, this is a good question. Once again, we work on it. Several groups work on it. I mean, it's maybe stabilization. It's not directly beta oxidation. I mean, it won't regulate beta oxidation. I can't believe that it will act on the complex of the mitochondria, make them respiratory complex, make them better. But probably it's the mitochondria itself. Maybe it protects mitochondria from mitophagy or some stuff, or it induces mitophagy, so there are less mitochondria or some stuff like this. It's more, I would say, something like that. And of course, if you have still your mitochondria, if they are still working but they are not affected with this process of autophagy, they will tend to react of triglyceride, and maybe they can use it better and make beta oxidation. At the point, I think it does not regulate directly beta oxidation, but it's an indirect phenomenon.
Okay, it's just to congratulate Jeremy, because I enjoy a lot with this fantastic talk. As Vlad said, it was a great opportunity to learn about this necroptosis that I think it could be very important in the future. And congratulations for your fantastic work. It's very complex to know if we can link for sure. When they appear the first drugs, antifibrotic drugs developed for the lung or for the kidney, we are with the temptation to include in the same pathway, but perhaps each organ show a completely different pathophysiology, and the mechanism could be quite different. At least the same noxus could play a different role in the different organs. And I think we need to keep analyzing separately. But I have no idea if necroptosis could be a common pathway in all these organs.
I mean, the same as you, so it's difficult to answer. But I mean, when you look at necroptosis fields, so I mean, it has been involved, for example, in atherosclerosis. So I mean, it was clear that if you have a macrophage going on with necroptosis, it will trigger lots of inflammation. It will contribute to increase the atheroma plaque. So, I mean, probably there is some link between vascular disease, but of course, I don't know yet.
Just speculating.
There are better scientists as me than in this field, but also for kidney. I mean, necroptosis has been involved in kidney death. So I mean, you could think that that's why it's quite important, this pathway, and it seems to play really a major role in lots of disease. And I mean, it's worth trying to inhibit it in patient or some kind of different disease. I think it's done in a neurovascular disease, and I think I would like to try it at least, and make lots of effort to prove that it's feasible and that it's more beneficial than adverse effect to push it at least.
I'm not. No, I'm not aware. And it's interesting to describe a mechanism of injury, but then if you want to move towards the therapeutic aspect, you need to make a drug. It's got to be druggable, because not everything is druggable, unfortunately. And it has to be in a way and a modality and a form that is acceptable for patients. And then it has to be highly specific for the liver. Because these mechanisms of cell death are ubiquitous, not only for NASH. So you may not want to interfere in other organs. So there are a lot of obstacles that are specific to drug development before you get to a disease, to a drug.
Yeah. Including the off-target effects.
And just in time, Vlad, and what about the inhibitors of specific caspase? For example, caspase 2, not just pan-caspase. Do you think we can test this type of drug?
Yeah, because caspase 2
I think
was beneficial, I think, in murine model. I mean, the inhibition of caspase 2 or the knockouts show some interesting results. So, yeah, I don't know if you are aware.
Perhaps when we talk about pan-caspase, it's not pan-caspase. It inhibits several caspase in different manners, and perhaps it's not the same to combine 3 and 4 than 2 and 1, or 2 alone. No?
Yeah, of course.
In animal models, it's worse. But I don't. You know, the gap between the result in the animal models to the humans is very, very difficult to feel.
And it's also
happening with the pan-caspase. But what is your opinion, Vlad?
I think it's difficult now that Emery Kazan failed to have a future in this direction. And if you ask Jeremy, he'll say it's not a good idea because it's going to block apoptosis, is going to trigger necroptosis. So, but then, as you say, it depends what level you block it and how much you block it. Is it 100%? Is it 50%? That could also play a role. So I don't know. Maybe it would be more interesting, I think, to see whether inhibitors of the inflammasome complex could be working or not.
Okay. Okay.
Yeah,
Okay.
Caspase-1. Yeah, probably. Specifically Caspase-1.
Okay, we have no more questions. I think we are on time to finish. Thank you very much again, Jeremy, Vlad, and Manuel for being here today. I hope you have enjoyed this time. And see you in the next edition of the liver seminars in September.
Yeah.
Congratulations, Jeremy. Congratulations, Javier. Fantastic liver seminar.
Yeah. Thanks a lot.
Have a good day.
Ciao.